Chapter 3, Segment 1. Neurophysiology is the study of the life processes of a neuron. Information flows within a neuron through electrical signals while information passes between neurons. All living cells have electrical charges. Neurons are more negative on the inside than the outside. An ion is an atom or molecule that has acquired an electrical charge by gaining or losing one or more electrons. A cation is a positively charged ion. An example would be potassium or sodium. An anion is a negatively charged ion, with examples being protein or chloride ions. All ions get dissolved in an intracellular fluid. Intracellular fluid is watery solution found within cells, also called the cytoplasm, while extracellular fluid is the fluid in the spaces in between cells and in the vascular system. The cell membrane is a lipid bilayer that ensheaths a cell and separates the intracellular and the extracellular fluid. A microelectrode is a small electrode used to record electrical potentials from living cells. This helped show that neurons are more negative on the inside. Resting membrane potential is a difference in electrical potential across the membrane of a nerve cell during an inactive period. A neuron at rest is approximately -50 to -80 microvolts, with a microvolt being a thousandth of a volt. The negative sign refers to the negative polarity of the cell's interior. Negative polarity is a negative electrical potential difference relative to a reference electrode. Inside the cell membrane is a lipid bilayer, which are two layers of fatty molecules where various proteins float around. The ion channel is a tube-like pore that allows specific ions to pass through the membrane. Some ion channels stay open all the time. Potassium ions can enter or exit ion channels freely, while the other ions are stopped by the membrane. This is called selective permeability. This is the property of a membrane that allows some substances to get through and others not to. The resting potential of a neuron reflects a balancing act between two opposing forces that drive potassium ions in and out of the neuron. The first being diffusion. Diffusion is the force that causes molecules of a substance to diffuse from regions of high concentrations to regions of low concentration. Molecules move down the concentration gradient until they are evenly distributed. The concentration gradient is a variation of the concentration of a substance within a region. The second part would be the electrostatic pressure, which is the ability of a charged molecule or ion to move via diffusion toward area with the opposite charge. Like charges repel while opposites attract. Cations are attracted to the cell's negative interior, while anions are repelled by the cell's interior and tend to exit to the extracellular fluid. The sodium-potassium pump is a mechanism that pushes sodium ions out of a cell and potassium ions in. This pumps 3 sodium ions out for every 2 potassium ions in. This consumes a lot of energy, and a large fraction of the brain's energy is used to maintain these ionic differences across neuronal membranes. The negative buildup caused by the cations leads to the exertion of electrostatic pressure to pull the positive potassium ions back into the cell. The concentration gradient and the electrostatic pressure eventually reach equilibrium with the cell's resting membrane potential at about -60 microvolts. The Nernst equation is an equation predicting the voltage needed to counterbalance the diffusion forces pushing an ion across a semi-permeable membrane. This predicts the resting membrane potential required to balance the high concentration of potassium ions inside the cell. Action potential is the brief but large charge changes in a neuronal polarization which are propagated at a high speed along the axon and presynaptic terminals. Hyperpolarization is the increase in the membrane potential, so the interior of a neuron becomes even more negative, brings the membrane potential farther away from zero. Depolarization is the reduction in the membrane potential, so the interior of the neuron becomes less negative, brings the membrane potential closer to zero. A hyperpolarizing stimulus is applying a hyperpolarized stimulus immediately produces a result. The greater the stimulus, the greater the response. This is known as a graded response, where the neurons change in potential. The capacitance is the membrane's ability to store electricity. Depolarizing pulses is the same procedure as a hyperpolarizing stimulus, unless the cell gets to -40 microvolts. This is called the threshold, and this is the stimulus intensity that is adequate enough to trigger an action potential. This makes the membrane more positive, a rapid reversal. Larger depolarizations produce more action potentials, not larger ones. The all or none property is the action potential is independent of the magnitude of the stimulus, so it either fires at full amplitude or not at all. Information is encoded in the frequency of action potentials, and an after potential is the positive or negative change in a membrane potential that may flow in action potential. Local potential is an electrical potential that is initiated by the stimulation at a specific site, which is a graded response that spreads passively across the cell membrane, decreasing in strength with time and distance.